Now we've got thousands right now of students who are learning this, who are maybe aren't ever going to go in as deep in the chemistry as you've gone. So what do you want to say to those people who are training in various medical fields and biology, in biology and these fields where they're going to take this and think that from what they read in their textbook that, hey, we've got the origin of life basically figured out?
You can still learn your medicine, you can still learn your biochemistry, you can still learn molecular interactions. So don't throw that away. But the statements regarding life's origin are weak and misleading.
I'm here with Dr. James Tour, and we're going to be talking about how our scientific professionals have been misled about the origin of life through information that's being given in college biology textbooks. Now, Dr. Tour is one of the top chemists in the world. He has over 800 research publications, been cited 100,000 times, has 200 patents. So he really is an expert in this. He's also a professor at Rice University. And thank you so much, Dr. Tour, for taking the time to be here.
Oh, my pleasure, Rebekah. It's very nice to be able to meet you.
Thank you. And I know you have spoken out so much about how the public is being misled through the media about the origin of life. And so today we're just going to be talking about one of the other ways the public is being misled, and that is through college biology textbooks. And we're just going to go through some of what this biology textbook says about the origin of life. And this is the most popular college biology textbook, Campbell Biology. And we'll be reading from the most recent edition that just came out in December 2025, so very up to date. This is used at prestigious universities such as MIT, Johns Hopkins, Harvard, Stanford, UC Berkeley, Cornell, and many others. So let's just get started with that. We're going to be talking through their section called Conditions on Early Earth Made the Origin of Life Possible. Do you have anything you want to say about that, Dr. Tour?
Yeah, the conditions on early Earth, the presumption on what the conditions were keep changing. In the 1950s, during the Miller-Urey experiments, those were run under reducing conditions. And that's what it was presumed that the early Earth had. And now the presumption is that they were oxidizing conditions, more like what we have today. So the jury is still out on what the conditions of early Earth were 4 billion years ago. And also, it doesn't really matter to me. I'll take it reducing or oxidizing. The barriers remain insurmountable, regardless of what the early Earth really was, the conditions.
Okay, well, they go on to say studies related to the volcanic atmosphere and alkaline vent hypotheses show that abiotic synthesis of organic molecules is possible under various conditions. Do you agree with that?
No, that's a bunch of baloney. This organic molecule synthesis coming out of vents, it's hard for us to know. You can't just measure what comes out of vents today because there's a lot of microorganisms that are degrade in there. But when you look at the conditions, actually, you have two kinds of vents. You have the ones that may be, I don't know, 125 degrees or something like that, and others that may be 350 degrees or hotter. So you have the two types of vents, some blowing out white smoke, some blowing out a black smoke. But it doesn't matter because the free energy for all of these molecules to polymerize is way too high. And so the free energy is positive. And so it can't work, and just heating it up doesn't work. You still have so much water around you. So with all that water, none of these polymers could form. Then if you look at the building blocks of the building blocks, which are the basic organic molecules, always racemization at those temperatures is much faster than polymerization. So if you had, and so all of our organic molecules have a stereogenic center. So for example, all the amino acids save one glycine, the simplest one, has a stereogenic center. All of those would be epimerizing. And you have to have had very high degrees of enantiomeric excess, like 99% plus. At the start of life. We know that because of chiral-induced spin selectivity, or else things would have heated up too much and become too hot in any living system if there was a disorder in that, and so we'd never get these molecules. So none of that has been shown. That's a bunch of nonsense.
Okay, well, I've seen people present papers and things that seem to be showing that, they've done these experiments and they think they can get it. So what's up with all these scientific papers?
Yeah, because nobody confronts them. I've said to the entire community, anybody with a PhD in synthetic organic chemistry or working in the area of origin of life or biochemistry, I will gladly engage with you. You take your favorite hypothesis on how these things may have formed, whether you want the RNA world hypothesis, RNA first, or if you want metabolism first, if you want proteins first, and we'll have a discussion. Come and talk with me about it. Nobody will come forward because, fine, you want to present this in a conference, go ahead, but you're speaking to a bunch of other people, and it's all an incestual sort of thing. Come and talk to me, and you know that none of this will hold up. So that's what I have to say.
Okay, thank you. Well, it goes on to say another source of organic molecules may have been meteorites. For example, fragments of the Murchison meteorite, a 4.5 billion year old rock that landed in Australia in 1969, contain more than 80 amino acids, some in large amounts.
Yes, you look at large amounts, it's still nanomoles per gram, number one. Number two, it might sound really exciting that you found 80 amino acids. The problem is you found 80 amino acids, and not just amino acids, many other organic compounds. You can't do chemistry on mixtures, cannot be done. And life has 20 amino acids, not 80 amino acids. So the other 60 amino acids are going to break this up and make it not life-friendly anymore. And it's not just amino acids. You had other carboxylic acids. You had other amines. All of those will start competing with each other. And so you can't do chemistry. You need more selective chemistry to be able to do this. And I'm not the only one saying this. Even John Sutherland, the great origin of life synthetic chemist, is saying that he says you need more constrained chemistry for this. You can't use what came on meteorites. So if you would like to contest with me, you have to contest with John Sutherland as well. And take those 80 amino acids in exact concentrations that you found on the Murchison meteorite and try to do anything with them in your modern laboratory, anything, and you will be unable to do that. So that there are 80 of them and all mixed together, now you're in a very hard place. And again, we're talking about nanograms per mole, or nanomoles per gram, actually. Very, very small quantities. So when they say in large amounts, it's large in whose mind? I mean, compared to what? And how many steps would it take to bring you to any living product? So you need more constrained chemistry for this. So there's a lot of things that argue against this being useful.
Well, I think that's what's confusing to the public too. It's confusing when people hear that, like, we've found these organic molecules, because that word kind of like makes it sound like life. So can you explain why, like, an organic molecule, like, how close is that to being life?
Yeah, it's not close at all to being life. I mean, some of these are relatively simple compounds. And so we have these techniques that have been around since the late 60s that allow us to detect down to even fundamentally one molecule. And so molecules, come in so many different structures. And then when they're mixed together, they're not useful. But you can certainly take very small molecules like ammonia, like carbon dioxide, like dinitrogen, and put them under certain conditions, highly explosive conditions and high heat conditions. And you can get structures that are reminiscent of the molecules that you find in life, but they're not useful. These are very simple amino acids, and they're very simple structures, and every one of these is racemic. You can't have racemic. You can't have the right-handed and the left-handed mixtures. So yeah, they are organic in the sense that they're made out of carbon, hydrogen, nitrogen, and oxygen, primarily with a little bit of sulfur and things like that. But so the definition of organic chemistry is that, yes, they're made out of carbon, hydrogen, nitrogen, and oxygen, whereas an older definition of organic means that it comes from life, but you can get a lot of these simple molecules. So you find water. I mean, water is the most abundant molecule in our body, but you don't have to have a living system to have water. You have water all over the place. And so just because you have molecules doesn't mean that life was there. What you'd have to have is stereogenicity. You'd have to be able to have high enantiomeric excess, and you'd have to have lots of copies of certain ones of them that are fairly complex. Now, again, these amino acids are very simple, small molecules. I mean, take something like a sugar, or take something more complex like a nucleotide. Very, very hard to find multiples of these with any stereochemical control. I know that what I have just said is hard for the common person to understand, and that's why I keep bringing it back to this. If this proved anything useful, you would think the experts would come forth and say, okay, Jim Tour, let me sit you down and explain this. But they don't. So even people like Steve Benner, I've offered him $10,000 to sit with me in front of a camera for 1 hour and explain to me how life could have come from these sorts of things. And he refuses. So you would think that if this were really figured out, these guys would just do it. They don't. So I know that what I'm speaking is hard for people to understand who haven't had an organic chemistry background, but it's telling that the experts themselves will not defend statements like this. If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
Okay, so this is from the section Abiotic Synthesis of Macromolecules, and it says a 2016 study demonstrated that one key step, the abiotic synthesis of RNA's two purine bases, adenine and guanine, can occur spontaneously from simple precursor molecules. The abiotic synthesis of the smaller cytosine and uracil bases was accomplished in 2009.
Okay, so these are the bases that attach on the ribose or deoxyribose and that make up the nucleotides. Yes, these bases are relatively simple compounds that can be made by the interaction of small molecules. I will definitely give them that. There's no stereogenic center on those, so you don't have any chirality problem with any of these small bases. So yes, these bases are aromatic in structure, and so they're ring structures, and they're very simple to get from smaller compounds. So I'll give them the bases. Yeah. And try hooking these bases now up to ribose or deoxyribose. Everything begins to change now.
Okay. So going on, it says, in addition, by dripping solutions of amino acids or RNA nucleotides on hot sand, clay, or rock, researchers have produced polymers of these molecules. These polymers formed spontaneously without the help of enzymes or ribosomes.
Okay, so what you can do is you can take amino acids and you can heat them up. You heat them up to like 125 degrees centigrade, which is the temperature that you heat things to destroy any life that would ever be there. and you dehydrate them, and you can get some hookup. But this is a bunch of nonsense. And the reason for that is that these amino acids, what they'll do is they'll take amino acids that don't have active side chains like glycine, and they will hook these up, so there's no side chain reactivity. But as soon as you start using the actual amino acids that you're going to need for real chemistry, about half of them have active side chains. So now the active side chains participate in the polymerization sites, and it's all bets are off now. Everything, it's game over for you. And so what they do is they take a preloaded amount, and then the largest ones they've ever seen are 14-mers. Generally, they might see tetramers, hexamers, octamers, but the largest one is a 14-mer. And remember, for amino acids to have any sort of activity, you've got to have about 100-mer. And they never see that because the free energy is positive. And the way they do this, they force it by dehydrating it. And in doing that, if there was a stereogenic center, then that epimerizes. At these temperatures, epimerization is faster than polymerization. They never want to discuss that. So when they look at their oligomers, either they were loaded so that they had no active side chains, or they never looked at the epimerization that occurred from the alpha carbon, the stereocenter. They never looked at that. When it comes to RNA nucleotides on hot sand, these things fall apart. RNA is a highly unstable molecule. And what you get is you get not just the 3', 5' linkage that you were supposed to. And this is taking a preloaded RNA nucleotide. So in other words, it's got the nucleobase, it's got the ribose molecule on there. So you bought that ribose, you bought the base already hooked together, and it had the activated phosphate group on it. So you have to have now not just regular RNA, but some activated group on there. And then what you do is you throw it onto your hot sand. Yes, of course you'll get coupling when you start evaporating the water, but you get not just the 3', 5', you get the 2', 5' coupling. Game over. And you also get branching from that 2' site. So again, game over. And these things will be very short. These things will be very short, but they're also not hooked up the right way. So none of the hookup is the right way. They're not telling you the truth. None of these could be active in a life-friendly scenario because all of them are improperly hooked together. Yes, they hook together, but they hook together to make garbage is what they do. Plus, you have no control of the stereocenters. You start heating things on the temperatures that they want, they're getting epimerization of these centers.
Okay, now that was all very technical, and I'm glad you went that technical for the people who are at that level. But can you break it down for a new biology student who reads this, and it sounds like they've taken some steps that are really close to, being able to synthesize life?
Yeah. So what they do is they buy amino acids from a chemical producer where they have them all. They have the stereochemistry, the chirality all confirmed. They throw them together on a hot rock. What's going to happen is all those stereogenic, or what are called chiral centers sometimes, are going to invert. They're going to epimerize, which means that they start equilibrating. And now what happens is they no longer have these polyamino acids that look like life. They have everything is scrambled. Plus, they can never make them very long because the back reaction is faster than the forward reaction. That's why they only get, the largest they'll get is 14-mers. The other problem is, remember your amino acids, half of them have a side chain that can also react. The side chain reacts, there's, for example, an amino group. It's just like the alpha amino group on an amino acid, but it's at a different position. They also have, branched side chains that have carboxylic acid groups. Those polymerize as well. So you don't get any clean polymers. No polymer that they form is life active. RNA, the RNA molecule that they use decomposes very rapidly, but RNA has a secondary site, and it's the 2' hydroxyl group. It is the site that has to be left free for that RNA to be active. That polymerizes just as well as the 3' site. Also, the RNA You can't just run off the phosphate as a typical nucleotide. You have to activate that phosphate. So you put on a human-made group on the end of that thing to get it to work. So all of this is loaded, and even with all the loading, what you get out is something that doesn't look at all like RNA and doesn't look at all like a polypeptide, because you have all the side chain reactivity. So they say you get polymers. You see what these polymers form. They produce polymers of these molecules. That is right. Polymers of these molecules. But are they polymers that are reminiscent of polypeptides? No. Are they polymers that are reminiscent of RNA? No. They just say polymers. Look, they didn't say we get polypeptides from the C terminus to the N terminus. They didn't say that we get RNA. They just said polymers. You heat up organic molecules, any organic molecules, you end up getting polymers. You get polymeric junk, and that's what they got. And that's why they said polymers. But they make you think that they got it. They got things that are reminiscent of polypeptides and RNA. It's smoke and mirrors. That's what they've done to you.
So from the section Protocells, it says recent experiments show that abiotically produced vesicles can exhibit certain properties of life, including simple reproduction and metabolism, as well as the maintenance of an internal chemical environment different from that of their surroundings.
Okay, so what they're doing is you can take, first of all, what they're doing is they're taking lipids that they bought from a chemical manufacturer that got these lipids from living cells, because it's very hard to make these lipid molecules, because you have to control the stereochemistry. So you start with a glycerol. The 1 and 3 hydroxyl groups are enantiotopic. You have to somehow distinguish these. Very hard to do this type of thing. Plus, many of the lipid long chains have stereocenters on them, whether that be easy stereochemistry or whether that be a sp3 stereochemistry. They have both of them on them. So you buy them. So you've bought these now, and they come from living cells. Now, when you take these molecules, you put them in water, particularly under shear. So you start putting them between rubbing forces, that can form a vesicle. A vesicle is a lipid bilayer. They naturally can form a lipid bilayer, but it's not cell-like, because the cell has so many different things. The outer portion of the vesicle has to be different than the inner portion of the vesicle in a living cell, because you have to have what's called a proton gradient. To have a proton gradient, you can't have the outside and the inside being the same. In all of their vesicles that they make, the inside and the outside are totally the same. Same molecules make up the inside and the outside. And the reason for that is because we have no idea how to make vesicles, synthetic vesicles, with control of the outside being different than the inside. So there's never going to be a proton gradient there.
Why do they say reproduction? Because that sounds like life.
Yeah. So this is something called the Marangoli effect. It's been around a long time. What you can do is you can take vesicles and you can start heating them up or putting them together under different conditions, and you can get more lipids, more of these lipids start inserting, and they'll grow and grow, and then at some point you hit a critical size and they break and they collapse to two smaller structures. And so this is common all the time. In fact, we do it in my lab. We work on these giant vesicles in my lab all the time. And you can put these under forces, and this is a typical Marangoli effect. It has nothing to do with life. These are just thermodynamic forces working on these molecules. Now, you say the inside is different than the outside. Yeah, now that you have this vesicle, now what's on the inside, you introduce something, things can't easily get in. So it's different on the inside than on the outside. But you're not going to have a typical proton gradient, and a vesicle, for anything that has to be living, has to have things that allow substrates and ions to move selectively through the membranes. None of this has any of those. You need transmembrane proteins that allow these sort of things. You need ionophores that allow these things to move in and out. These have to be highly selective. So yes, you can get something, you can get a vesicle, but that is nowhere near life. And when they say reproduction, they just mean that it can grow and grow as you put in more of the lipids. These lipids will insert, it hits a critical size, and then it collapses and it goes to two smaller ones. This has nothing to do with life, and it's been seen all the time. So for example, if I take a drop of water, I put it on a table, that drop of water tends to form a circular pattern. Why does it form a circular pattern on my desktop? Why doesn't it form a star-shaped pattern? Or why doesn't it form a smiley face pattern? No, it forms a circular pattern because it reduces it goes to a minimal disruptive surface. It goes to what's a thermodynamic most relaxed state. So it will drop in its energy and go to a spherical structure. This is where thermodynamics are pushing something to attain a spherical structure. Here you have thermodynamics that are allowing a critical size to form, and then it breaks into smaller ones. So if you keep trying to add water to that droplet on your table, at some point that droplet will split into a couple of different droplets rather than staying as one droplet to minimize the energy of the system. And that's exactly what's happening with this. It's just this very simple little elementary school experiment, and they're claiming that this is the reproduction, and they're trying to use these words. And then when they say simple reproduction and metabolism, there is no metabolism. There's no metabolism. It's not consuming anything and making any energy out of that. So this too is nonsense, Rebekah.
Thank you for clarifying. So they show a picture of the vesicles here. It says vesicles can divide on their own, as in this vesicle giving birth to smaller vesicles. This vesicle has incorporated montmorillonite clay particles coated with RNA.
Yeah. So this is exactly what I just told you. You can keep putting in lipids, and it'll keep growing, and then it hits some critical size, and then to minimize the energy, it will collapse into smaller vesicles. That is a purely thermodynamic effect. And you say that yes, if you put RNA in there, montmorillonite clay is a sticky clay, meaning that it has a surface charge that RNA will be binding to. And so yes, these vesicles will engulf anything that happens to be around them. And so they'll engulf it, and they'll say, well, we got RNA in there. First of all, you bought that RNA. That RNA was made in a living cell, and you bought it from a chemical manufacturer. You didn't find that thing. You didn't find it on the Murchison meteorite. There is no RNA in the Murchison meteorite. And so you had to buy that. But it will engulf anything that happens to be around it, and you have little particles of clay, this thing will engulf it. I mean, I can take little particles of plastic, and this thing will engulf it. There's nothing magical about particles of clay with RNA. And so again, this is nonsense. I mean, anything that you throw in there, it will engulf. If you have a little fountain and you throw a coin in there for good luck, yeah, the coin is in there. Hey, look at that. It just engulfed that coin. Yeah, well, I threw it in there. I put it in there, and this is what they say, it will engulf it. I mean, this is all that they're saying. I mean, you could take a little droplet of water on your desktop and drop some little particle inside that drop of water. You say, whoa, it engulfed it. Yeah, well, you drop that on it, or you can have a little particle on your desktop. You can push this little drop of water over it. It'll engulf that little particle. So there's thermodynamic surface tension forces that will do this. On this, it's got the thermodynamic forces of the lipid bilayer. So again, this has nothing to do with life. Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org/donate. All U.S. donations are tax deductible. Thank you so much.
Well, what about from this section? It says it's called self-replicating RNA. It says natural selection on the molecular level has produced ribozymes capable of self-replication in the laboratory. Thus, life as we know it may have been preceded by an RNA world in which small RNA molecules were about to replicate and store to genetic information about the vesicles that carried them.
Nonsense, pure and utter nonsense. So you can look at the work of Gerald Joyce. He has worked so hard to try to get RNA to reproduce itself. And up until a few years ago, you could never even get more than 10% of the RNA. Now he's come out and he's saying that he can get these short RNAs to reproduce themselves. But he's got so many human-made steps to push this thing. He's got ligating steps where you're taking two strands and hooking them together. We covered this on my YouTube channel, Dr. James Tour. This was in a discussion with Rob Statler, who went through this, and he took the latest work of Gerald Joyce and showed all the human work that you had to go through to try to get some sort of replication. There is no self-replication. There is none. In the laboratory, you don't just throw this together. It was about 20 different steps of pushing this thing and putting it under certain conditions to get it. And even then, the yield was so low. And so then they extrapolate this. They say, see, RNA, and it has genetic information. What information? There is no informational code. This is like my taking a box of letters. So say I took a book and I cut that book up into individual letters. And I put those letters in a box and I shook them all up. There is no code there. There's no code anymore when I've broken this thing up. Now, you keep shaking and shaking, you might get 3 letters to line up next to each other that form a very small word. But that like sit or something, but that won't be useful because now it's surrounded by other words that mean absolutely nothing. There is no genetic information. This code problem is a huge problem. And what this statement in this book has done is to presuppose that somehow it found a genetic code, and the genetic code came from the vesicle that carried them. No, it doesn't happen that way. What vesicle that carried? There's no information in that vesicle. There's nothing in that vesicle. And especially the vesicles that they make, which are all the same lipids inside and out. There's no informational code there. And this RNA world, if you had a molecule, one RNA molecule that happened to have a particular configuration, that would last hours. In stabilized water, it would last hours. In typical water, where you have divalent ions like magnesium, it would last minutes. That's it. So there's not enough time for any chemistry to take place. So this too, Rebekah, is nonsense. And I would challenge the authors of this textbook to come and sit with me and explain this to me and then start going through the chemistry. They will not. They will not, because they know it is a bunch of nonsense.
Well, and that's the thing, is that when we read these textbooks, we think we're getting facts. And so it's really hard to separate fact from fiction when you don't have all the information, you don't have the knowledge of chemistry that you have. So like, what's going on that these textbooks are printing things that are so grossly inaccurate?
Yeah, they're getting this thing from Origin of Life researchers that wish that they had this stuff, but the Origin of Life researchers themselves will not defend this. There was another very rich man that tried to get me together with 5 Origin of Life researchers, and they all refused to go. Even no filming, no attribution, just a discussion on where we need to move this field. And one of them said, "Well, I would only come if I didn't have to defend anything that I've written." Anything that he himself has written, not that Jim Tour has written, that he'd have to defend. He refused to defend anything that he himself has written. So yeah, they're feeding us a bunch of nonsense. They really are, and that's why in the end, I'm going to come out looking really good on this. I'm not sure that I'm going to live long enough for that to happen, but all of this is going to go away. It's going to go away. I mean, this is like nonsense sometimes that we read historically, things that people believed, but it's going to go away.
I hope you're right. So, continuing on with the self-replicating RNA, it says recently, Dr. Jack Sostak and colleagues at Harvard University succeeded in building a vesicle in which copying of a template strand of RNA could occur, a key step toward constructing a vesicle with self-replicating RNA.
Yeah, if you throw in enzymes, when you throw in active enzymes that you've taken, first of all, you start with RNA already, and so he starts with a template strand of RNA. So where did he get that RNA? It came from a living cell. Then you buy other nucleotides. Where did you get those nucleotides? You bought them. So they didn't occur on their own. And then you add enzymes to that within a vesicle, and the RNA begins to duplicate itself. Well, whoop, this is what nature does. This is what natural systems do. They have enzymes that do this construction. This doesn't happen on its own. And Jack Sostak knows it. This is why Jack Sostak won't engage with me. He's not at Harvard anymore. He's moved to University of Chicago. A very, very nice man, but he will not defend his own work. He will not. He even said in 2014 that he would have life in the lab in 3 to 5 years, more like 3 years rather than 5. He's missed his deadline. He's never made life. And then a year later, rather than saying he'll have life in his lab, he says, well, we're still having trouble making the RNA. So it depends what kind of audience he's talking to. When he's speaking to professionals, he has trouble making the RNA. When he speaks to the general public, he says he's within 3 to 5 years of making life. That's part of the saga of the nonsense that gets thrown out there.
Well, that's what's also very confusing, is because right now, if you Google, have people made synthetic cells, have scientists made synthetic cells, it'll pop up and say, oh, yes, we've created cells in the lab. And so it's very confusing. Now, if you keep reading on, it'll kind of clarify that it's not, life from scratch. But I mean, it sounds like a lot of people that I talk to do believe that we have created cells.
No, nowhere close. So what they did, a synthetic cell, is they took a living cell and they copied the genome, and then they took out the one that was there and they put the copy in. And so it started running again. That's like taking the computer chip out of your car and putting in a new one, and the car runs again. Or they've taken the genome and they've knocked many portions of it out so that it can't function anymore, and the cell keeps running. But now it's got a modified genome, and they call that a synthetic cell. But all the structure was already there. We have no idea how to build a cell.
About billions of parts or millions of parts.
Billions.
Billions that they're keeping in place, that are alive.
Yeah, and those parts have orientations between them that have substructure arrangements that are 1 in a billion. I mean, it's just so hard to think about putting something together like that. So yeah, it's nonsense. You have anything else for me? Any other statements?
Oh, yes. Once RNA sequences that carried genetic information appeared in protocells, many additional changes would have been possible. For example, RNA could have provided the template on which DNA nucleotides were assembled.
Okay, we still don't have the the ribose to build the RNA. We still don't show the nucleobases, the 4 nucleobases hooking to the ribose. We still don't have the active phosphate group on there. So none of those parts of the building blocks are the building blocks. Now, he still has not made for us RNA, because remember, any synthesis of RNA gives junk. It gives polymers that aren't the right shape. So what he's doing, he's saying, let's just say we had RNA and it has the genetic code. What do you mean genetic code? I mean, if it just formed randomly, there is no genetic code. There's a huge presupposition here that you're getting the life-friendly information here. And there's no genetic code there. It's just a bunch of random letters. But say you did have one, and say you did have it in a vesicle, and then it would have been possible, for example, that RNA could have started making a template for DNA. Well, no. RNA with nucleotides of deoxyribose nucleic acid, the nucleotides of those, now you have to have a different base there. So one of those 4 bases has to have a different structure. And so why were none of those around when the RNA was made? So all of a sudden, you've got your extra base there, and you've pulled out one of the bases. So you've pulled out one, and you've put in another. And now what happens is you have to get these hooked up. But without associated enzymes, this has never been shown. RNA does not form DNA unless you have many associated enzymes. So this doesn't happen. Go ahead, read the next sentence.
Okay, once DNA appeared, the stage was set for a blossoming of new forms of life, a change we see documented in the fossil record.
No, wrong, lie. This is not true, because DNA by itself does not do anything without associated enzymes. DNA has to make RNA through templates, but you have to have all the RNA nucleotides present that were not present when the DNA formed, by the way. So they magically disappeared, and now they've reappeared to now make this. And you have to have all these associated enzymes, but the enzymes have never formed yet. There's no enzymes. So DNA can't make RNA without a host of associated enzymes to bring all these nucleic acids into alignment and to make this happen. So that too is nonsense, and none of this is shown in the fossil record. None of it. So this is all nonsense. Go ahead, read the next one.
Well, in changing from an RNA world to today's DNA world, genetic information must have flowed from RNA to DNA. Now, this is where they want the student to respond, and they say, suggest how it could have occurred.
Yeah, it could have occurred if you got rid of all the RNA nucleotides and now just brought in DNA nucleotides magically. If you could have made the DNA nucleotides, which means you have to have made deoxyribose, and you have to have made then all the associated enzymes. So the answer they want is DNA, the RNA templated the DNA, and the DNA then took over. But the template formation does not work without a host of associated enzymes. That's what they're not telling you. So everything you've presented to me, Rebekah, is just nonsense. And so we've showed exactly what I've been saying, that these textbooks are teaching wrongly. And yeah, I don't know, I don't know how much more I can cry this from the housetop that this is a real problem, and I think other people will start crying out with me. But those experts in the field are not willing to defend any of the statements that you just read to me in the book. The experts won't confirm any of these.
Well, now we've got thousands right now of students who are learning this, who aren't ever going to go in as deep in the chemistry as you've gone. So what do you want to say to those people who are training in various medical fields and biology, in biology and these fields where they're going to take this and think that from what they read in their textbook that, hey, we've got the origin of life basically figured out.
Yeah, you can still learn your medicine, you can still learn your biochemistry, you can still learn molecular interactions. So don't throw that away. But the statements regarding life's origin are weak and misleading. And so just skip over that because it is a bunch of nonsense. But the other things you learn in biology about how molecules interact, how molecules work together, mechanisms of things, those are well worth studying because they'll give you a host of information upon which to build. But it is just in this area of the origin of life, these statements that we read are utter nonsense. So we don't know where life came from. That does not mean that we should not study. That does not mean that we should give up the study of biochemistry and organic chemistry. It just means that we don't know how life first occurred. But we can learn a lot about how to sustain life, how to make it better, how to fight diseases. And that's what we want to learn and continue to learn. So I encourage students to go into those areas.
Thank you so much, Dr. Tour. Thanks for your time today. Is there anything else that you wanted to share?
No, I think that's about it. Thank you for the work that you do, Rebekah, and hopefully this will make it clear to certain people. And I invite if anybody feels that I'm wrong on this, I can't engage all the YouTubers and all the people that don't know anything about chemistry. I haven't time for that because there's so much that they don't know and they don't understand. But if anybody among your listeners has a PhD in organic chemistry or in biochemistry, and they'd like to come on and contest with me, I'm sure you'd invite them onto your channel, but and then bring the two of us on and let us have a discussion. And or if you'd like to come on my channel, Dr. James Tour, we'll have a discussion there. Or we could go on some other neutral channel and have a discussion there. Or I'll come on your channel and we'll have a discussion there. But you have to have a PhD in organic chemistry or in biochemistry or working in the area of origin of life, because without that, I don't have any hook to really get firmly into a deep conversation with you, because you just wouldn't understand the chemistry. It's not that I'm trying to be conceited. It's just that we're just saying you have to learn a language. You have to be able to learn the vocabulary of a language before you can begin to speak it. And that's what we're saying. And then along with that, that's all I have to say. Thank you, Rebekah.
Thank you so much, Dr. Tour, and thank you for all the great work that you're doing in science and for the Lord.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.